All right, let's start. I don't think it's on freeze. Awesome. So, let's start today with our idea as always. This one is just our weekly check-in. It's the first time we guys this week. So we'll move on to that, and then we'll talk about what we're doing for today's lesson. So, as always, we start with a nice little Punnett, so that way we have some time just to chill before we start our work. Cool. So I'm going to randomly pick the bottom of my chart. As always, I think it's nice just to share. The random question for this week is about best friends with celebrities. Who would you like to have as your best friend and why? And I'm going to pick two people out, so hopefully you're ready to share. First one sharing is Mitchell. So Mitchell, who is a celebrity that you would want to be friends with? A celebrity you'd like to be friends with? Don't know? Okay. I don't think I know who I want as my best friend celebrity. Hopefully the second person has an answer. How about you? Kevin Hart. Why?
Because he's funny and he inspires other people.
I like that. He's funny and he inspires other people. You're the first person that said Kevin Hart. I've seen a lot of people say actually Kevin Hart as their kind of celebrity best friend. Cool. Next one is micro goal. So the person that's sharing their micro goal for this week is going to be Brooklyn. What's a goal that you have for this week? You know, it's really good for us to have goals, so that way we can make sure that we're on track. All right, I'll pick one more person out, and that's going to be Neil. What's your goal for this week? Wake early. I like it. You get a lot more done when you guys wake up early. But do make sure you're going to bed at a reasonable time. You get enough sleep. You guys have growing brains. Yeah? Right. Anything that you want to share before we move on? Anything really interesting over their weekend? Things like that? No? I went to a wedding over the weekend. That's my plan. It was 90 people because with Red you can't have them over 100, and you have to factor in staff as well. It was up in Omaha, which is up north, and it was a nice little kind of beach kind of weekend away. So it was really recharging. So I figured I'd share that with you guys. That's what I ended up doing. All right, ready to switch gears? Okay. So we're switching gears and thinking about the knowledge that we're going to be building together. So just to recap what we did last week, we looked at DNA. And we looked how that DNA links into phenotype. So it's DNA, protein, and then phenotype, which is our traits. We also looked at how mutations can cause those new traits. Really interesting kind of stuff to think about. Cool. So today, what we're going to be building on the next vocabulary, and we're going to be using those Punnett squares. And with those Punnett squares, we're only going to be doing mono-hybrid cross. I'm specifying now the type of Punnett square you are doing, because you're also going to be doing dihybrid crosses as well. So mono meaning one, di meaning two. So we're basically doing lots of review from last year. This is all the stuff that you've hopefully done before. And it's to build up for the new material. Like I said, dihybrid crosses, we're able to look at two traits at once and see how they're inherited. So hopefully an easier day and stuff that you guys have done before. If you guys need the learning session, is on the board over here. So we'll be switching it. I do recommend that you write down the date and the learning session to your notes, so that we can keep track of what we've covered. I suggest either paper or you can do it online. I'm not too fussed. And then also, since this is the week, I thought it's nice to share a weekly episode. So in this case, we are thinking about while the Motorola grub is small, it can take through the white pine, which basically, if you're thinking about our kind of statements, they're power in small things. So power in small things, I like that one because I'm quite short. I hope I'm powerful. But also thinking about your actions. Lots of small actions can have lots of big impact. So do kind of keep that in mind for this week. All right. Let's talk about the lesson plan today. Lots of cover time for you guys to process, and then a little bit of me giving you notes. We'll start with the monkey choice. I'll give you a choice of either doing the DNA bracelet that we didn't have time to finish last week, or you can do some side pad work. I'm not fussed which one you do. Then we'll talk about some vocabulary and how sex information works. We'll do describe and explain. So you guys can start exploring. Then we will look at some more vocabulary and Punnett Squares. And then I have a consider the following question. We have diseases from last week that I just want to talk about. So a lot of you guys are asking me about how they're inherited. And then we'll finish with more multiple choice so you guys have some chance to process what we've talked about so far. Okay. All right, phones off the table. Presley, phone off the table. Don't want that distraction. Okay, from our success criteria, we're jumping around a little bit because just keep that in mind. From the success criteria, we're not necessarily going to do them in order. So what we're looking at for today is knowing those terms genotype and phenotype. Genetics has a lot of vocabulary. We want to make sure we use the right one. We're also going to be using Punnett Squares to predict results of a crop, and that should be refreshed from last year. We're only doing, like I said, monohybrid, which is just the one trait, and then we're also going to be defining the term pure breeding. Questions with those? Keep going. All right. So let me explain your multiple choice. So you guys remember, not from my little jump. If you guys remember from last week, we were talking about the way DNA codes for proteins and that those specific traits of those proteins give that trait. In order for DNA to do that, what it does is the sequence is read as a triplet, and that triplet then says this is the amino acid to grab. And when we have that sequence of amino acids in the right order, it can then fold into the correct protein. So I hope reinforce the idea. I thought it might be fun to make a DNA bracelet. Some of you guys started this the other week, but I was thinking about it and I was like, you know what, let's do full names. Because I did my full name, and that was like long enough to actually make a bracelet. So that's why I want you guys to do full names. And when you do that, you're going to look for the letter. You're going to then grab the letter, figure out what the codon code is for that. And then you'll be able to get some of the little moon rubber bands and put them in order for your name. So that's option number 1, if you guys want to do that. Option number 2 is some SciPad pages looking at mutation, and in a moment we'll pass out those SciPad pages. Or not SciPad, the SciPads. So I talked to the other biology teacher, ladies. I talked to the other biology teacher, and she said that it's better for you guys to have the full external booklet because you need some of that cell material in order to do the microscope assessment. So I'm just going to issue out the full SciPads for you guys. So let me show you what the first choice of your bracelet is, and then you guys can choose if you want to do the bracelet or if you want to do the SciPad pages. Okay. So if you want to do the bracelet activity, what you're going to do is you're going to take your full name and you're translating it into the DNA code. Proteins carry. So if you guys look here, the type is AAG. So I put AAG, and then I've taken the E, so that's GAG, and then I look for the R, and that's CGT. So I've written that down, and then the I is ATA. So I've done my full name, so that way we have enough to make a bracelet. So I've done Carrie. This is how tan my middle name translates into, and then this is garlic, how my last name translates into. Then what you need to think about is grabbing the right colors for the different letters. So every A I have, I should have red. Every G I have, I should grab purple. Every C is going to be, you need to choose pink or blue. I'm not fussed which one you want to do. And then T's are going to be G. Remember, with the pink or blue, pick one. So if we look at my bracelet here, you can see I have red for the A, another red for the A, and then I have purple for the G, and then I have another G, so that's purple. And what this whole bracelet translates into is my name. So it's a nice little blue band. So that's option number 1 if you guys want to do that as your activity. Option number 2 is some SciPad pages just to review those mutations. Sound good? All right. You guys are welcome to listen to music while you're working on this first Mahi choice. I'll pass out these if you guys want to do your bracelets, and we'll pass out the side pads as well. If you finish early, you can start working on the side pad pages. Sound good? Right. So, I know some of you guys are still working on your bracelets, but most of you guys have finished. So what I'm going to do is I'm going to move on to the notes. Those that are still working on your bracelet, you can listen and keep working on your bracelet. Side pads can be closed because I'm going to just, like I said, go through some of the notes. And what else did I want to do? I think that's mostly it. So return to your seats. Get ready for notes. Jen, Cass, Presley, back to your seats. Oh, yeah, your friends sit there. All right, go back to your seats so we can start doing some notes. Yeah? No, she needs to come back. She needs to do notes. All right. What I want to talk about for today is some of the new vocabulary. And some Punnett Squares. Well, no, plenty of half an hour. Okay. So, all right. Slowing our walker down. Come on. Oh. Notes. In your printer? Do you have a booklet? All right. If you don't, I have some spare copies. Okay, so the first thing I'm going to talk about when we're looking at vocabulary are the genes and the allele and the genotype. So, oh wait. All right, so we are talking about genes. One thing that gene number refers to a specific section of the DNA, and that sequence can be different. With that sequence being different, that is the result of things like mutations. So mutations can occur, and it changes the sequence so that way you have two versions of that gene. You will receive two alleles, one from each parent. So male, female, you get one from each, mother, father. The combination of the alleles that you get is known as the genotype. So we talk about phenotype. Phenotype is physical. Genotype is the gene. About what two alleles do you have? Depending on an organism, depending on what you have as your two alleles for those genotypes, that will determine your phenotype and what physical trait you have. Cool. I think that's all I want to say for that so far. Are we okay with those terminologies? Alleles, genes, phenotype, that sort of stuff. Yeah? All right. So to start us off with those Punnett squares, the first one I'm going to look at is sex determination, because it's nice and easy. It's also very unique. So this is found obviously in humans. It's also found in most mammals and even insects. So a lot of different animals will use this method as their sex determination. For humans, we have 46 chromosomes. 44 are known as autosomes, and then 2 are your sex chromosomes. Obviously, you get one from mom, one from dad. Females are XX, males are XY. This will hopefully be, like I said, revision from last year. Females only have that one chromosome to give. So females have to give an X chromosome. Males can give an X or a Y. So that means that since they can give X or Y, they determine the gender of the baby. I should say the sex of the baby. So let's look at how this works with our ratio. I'll just grab my document camera and a piece of paper so we can see it, how it plays out. Yes, very simple stuff. XX, XY. So just going to refresh our Punnett squares, and this is a nice easy Punnett square. Cool. And I think this one is in your booklet, so if you need space to write your Punnett squares, you do.
In your books, it's in the booklet itself. What is it? A whatever. It's those little booklets that are these guys. So there should be a page in there for it. Yes, there's a little slide in there for you. All right, so determining components, we have X and we have Y. So females are XX, males have the XY. And I'm doing it 2 different colors so that way it's easy to kind of tie which one's father, which one's mother. With Punnett squares, remember you are pulling the top one down. So females only have the choice of giving an X chromosome. With the one on the side, you're pulling it across. That one's going across that way. And that's my Punnett square. How do Punnett square I can make predictions? I can make predictions based off the genotype and off the phenotype. Genotype is referring to what 2 alleles that I have. So when I look at that, that's 1, 2 that are XX and 1, 2 that are XY. You can also say 50% XX and 50% XY. All these are ways to communicate across what we see as the genotype ratio. Phenotype is the physical trait. So we're thinking male and female. So if I have XX, what am I? 2 female, 2 girls. What about males? 2 as well. It's really important that you guys can do these ratios because we're going to use that to help justify what's going on. You say 50% female and 50% male. Let's shift that down. So that's what we see for our genotypes. Are we good with that? Can I move it? All right. So with that, of having a boy, 50% having a girl. So it's an even ratio. The other thing that's important to remember is that the previous fertilizations do not affect the outcome for the future generations or future offspring. So let's say someone has had 4 boys and they have a 5th child. What's the likeliness that they're going to get a boy again? Still 50-50. So each one of those fertilizations is a separate event. So even though you've had previous siblings that are all boys, it doesn't necessarily guarantee that you're going to get a boy again. Cool? Make sense? All right. Here's a little joke for us. So if you guys remember Henry VIII, he was very obsessed with getting a male because he needed a male heir. And he blamed the females for giving him daughters. He beheaded them and divorced them. But whose fault was it that he wasn't having any boys? His fault, because he determines the gender. Yeah? All right. Cool. Moving on to our next task is our describe then explain. So we are thinking about the discovery of genetic inheritance, and we're thinking about Gregor Mendel. He was a really famous friar. I'll wait till you guys are done talking. Still some chit chat. Presley, go on. So what I want you guys to do is make some observations on how he figured out genetic inheritance. So in your booklets, you have a hopefully colored photocopy. I don't have a colored photocopy here. And I want you guys to make some observations and draw some conclusions on it. That's what describe and explain is for. So some things to keep in mind is that peas have one gene that control the pod color, and the 2 color choices are either green or yellow. The generation up here, I'll get my little laser pointer. This generation is the parent generation. They are purebred. This one's green, and this one's yellow. And when you cross those 2 together, you get only green. What they then did in the next generation is they took the ones in the first generation and they crossed them together. And from that, they have the third generation, or the the F2 generation. Parent, first, second. And in this new one, there are 3 greens and 1 yellow. So I want you guys to think about this image that I provided, and I want you to describe what you see and explain it as well. So there's space in the little booklet. Let me just show you the booklet again. And that lighter color is yellow, if it's not. So I want you to make some observations, describe what you see. You can write the description down here, or you can write the description on the image itself. And I want you to also explain why you think you see that pattern. Give that a go, and then we'll talk about it. What we see in those images. Cool. So, what I was hoping, I'll wait till Lily to know. Ready? All right. So, what I was hoping you guys would realize from looking at this example was that you were going to recall some of the information you have on alleles and hopefully the dominant recessive ideas that have come back to you from last year. So, some things to think about when we're thinking about the alleles is, don't have my clicker plugged in. So when you're thinking about alleles, and we're thinking about what we learned last year, we learned about the terms dominant and recessive. So dominant is talking about how it's always going to be expressed. So if I have inherited one of those alleles, then I will express that trait. I want you guys to be really careful when you're explaining what dominant means, that you're not using the word more common, because it may not necessarily be common, and we'll see that with the population genetics. And I don't want you to describe it as being stronger, because it's not like a strength thing. It's just expression. So you're just stating that it is expressed. Recessive, on the other hand, is only expressed if the dominant allele is not present. That's the key kind of thing. So, I have to have 2 recessive alleles in order to get the recessive trait. Therefore, you can only get that recessive trait if you have a homozygous recessive genotype. And we'll refresh that as well for our ideas. Again, do not call it the less common or the weaker, because it may not be less common. That recessive allele might be really, really common in the population. It's not weaker, it's just not going to be expressed if that dominant one's there. Cool. So, I'm mindful of the time, so we might just kind of talk about this through it. So when we looked at the Mendel-Pease examples, we had the green and we have the yellow. Which of those 2 traits do you think was the dominant trait? The green. And which one was the recessive? The yellow. And the reason why has to do with how we see the generational inheritance. So in that first parent generation, we had green and we had yellow, and then when they bred together, we only got green. Yeah? So that tells me that everybody has inherited one of the dominant alleles, and so all the offspring have the dominant trait. When I then bred together the first generation, the 2 green pods, these guys had 1 yellow and 3 green. So what this tells me is that the recessive trait is now able to come through again. So it's been skipping a generation. So again, with that recessive trait, it needed to inherit 2 recessive alleles to do that. All right. So, genotype versus phenotype. There are 3 different types of genotypes that you guys would have talked about last year. First one is homozygous dominant. So you have 2 of the dominant alleles. The second one is homozygous recessive. So you have 2 of the recessive alleles. Homozygous is just referring to having 2 of the same. Think homosexual. Heterozygous means you have 1 dominant and 1 recessive allele. So you have a mixture of 2, but because you have at least 1 dominant one, you're going to get the dominant trait with a heterozygous. Again, heterozygous, think heterosexual. Cool? All the vocabulary ties together. So when we're looking at the Mendel-Pease, we had, do you think, let's go back to Mendel. Do you think this green pea at the top with the parent generation, is that one a homozygous? dominant, or do you think it's heterozygous? Homozygous dominant. And how do you know that?
Because the offspring was.
All the offspring were green. So that means everybody got a dominant allele. This case here, though, I can look at it and I see it must have gotten one dominant one to be green. It must have gotten one recessive from the yellow parent. And so these in the second generation are all heterozygous. And the two heterozygous, when they breed together, we get a 3-to-1 ratio. So that's a real good hint that you've crossed two heterozygous together. Are we good? All right, I'm just going to skip it ahead. Cool. So, purebred is another word you guys need to know. So, which two genotypes are the purebred genotypes?
The dominant.
The dominant or the recessive homozygous. So, they're considered pure breeding because they only have the two of the same alleles. If I have two different alleles, like the heterozygous, I'm not pure breeding. No, because you're a mixture. All right. Cool. So either homozygous dominant or homozygous recessive, you basically need two of the same alleles. So was the first generation a purebred generation? The parents. The parents? Yes, the parents were. Were the second ones? No. Second generation. No, they weren't. They're heterozygous. All right. So let's do some Punnett squares as revision. We're going to do eye color as the example. So we're going to do brown eyes and blue eyes to keep it nice and simple. The other thing to remember when we're talking about Punnett squares is that uppercase letters are referring to dominant and lowercase letters refer to recessive. Again, let me just restart my camera because it seems to not like it when I unplug it. Let me just go to the PowerPoint so I can see. All right. So we'll do a heterozygous cross, and then we'll do a heterozygous homozygous cross. All right. Cool. And I will scan these notes so you guys feel like you need them. So for practice. So I said I have both heterozygous parents, and we're doing eye color. So capital E, lowercase e, capital E, and lowercase e. So what color, if brown is the dominant, what color should both my parents have for eyes? They should both be brown, right? And I think you guys have space in your booklets for this as well. So you can write that down. All right, remember we are pulling the letters across or down. So in this case, I'm pulling this capital E down. I'm pulling this lowercase e down. Doesn't matter what order you write them in, but I tend to like to write the capital letter first and the lowercase second. All right, then I'm pulling these guys across. And that's my Punnett square done. This ring a bell? All right, let's now, Presley. We'll do the genotype ratio first. So how many capital E's, the homozygous dominant, do I have? Just the one for capital E, capital E. How many capital E, lowercase e's do I have? 2. Oh, sorry. 2. All right, how many little e's do I have? Just the one. Cool. So I have 25%, I have 50%, and I have another 25% there. Are we good? All right, phenotype ratio. So we're talking about traits with the phenotype ratio. How many would express the brown eyes? 3. 1, 2, 3. So we have to keep in mind that we are adding these two together because they're both having the same physical trait. So I would have 3 brown and 1 blue. Oops, I was going to write brownie. And that 1 blue. Does that make sense? Bonnie, you look kind of confused. Are you okay? Yes. Is this okay? Do you want me to review anything? No? All right. One more example, or are you guys okay? You okay? Okay, I can skip ahead. If you guys want, what do we got left? We have about 5 minutes. We're just trying to see what else we have on our agenda. I'll quickly finish this stuff and then if I had a cross and all the offspring had brown eyes, and is it the same as my predicted ratio that I just did? If all the offspring, I had 4 children, and all 4 children had brown eyes, is it the same as what we predicted in the ratio? No, because in the ratio we predicted 3 to 1. So in theory, I should have 3 with brown eyes and 1 with blue eyes. What went wrong? Is my baby daddy not my baby daddy? No. It has to do with sample size. So that's something else to remember as one of your justifications. So predicted ratio and actual ratio may not match. And the reason why it may not match is because the sample size is too small. So is a sample size of 4 offspring large enough to see the ratio? Probably not. So think about it when you flip a coin. If you only flip a coin twice, you may not get one that's heads and one that's tails. But if you flip it 1,000 times, it should more or less fit that 50-50 ratio. All right. So thinking about those diseases from last week, we watched some videos on Huntington's, and we watched some videos on cystic fibrosis. I already told you the answer. Anyway, that was silly of me. All right. Huntington's disease, the one where the woman described the video, that is an example of a disease caused by a dominant allele. Cystic fibrosis is caused by a recessive allele. So if we're thinking about these discussion questions, which disease do you think is likely to skip a generation? The cystic fibrosis or the Huntington's? Which of these 2 diseases? Huntington's is a dominant allele, cystic fibrosis is a recessive allele. So, which of these 2 diseases do you think skips a generation? The recessive allele. So cystic fibrosis skips generations. So you may not have cystic fibrosis, but your offspring do. And we saw that in that video from last week. Which of these two diseases is, if your parent has it, you have a 50% chance of having it too?
Which one of these 2 diseases?
That if your parent has it, you have a 50-50 chance of having it too. The Huntington's, because it's a dominant allele. So if your parents have Huntington's, they have a 50-50 chance of passing that defective allele onward. Sickle cell anemia, we're going to hold off on. That was a little bit more complicated as part of the new level 2 stuff that you're learning. Okay? So, just as a quick little wrap up, there is the Mahi choice if you want to do some more follow up at home. So, great work today. I'll give you guys some coffee beans. So today you guys should know how to use more genetic vocabulary, and you should be able to do Punnett Squares. You should be able to do the genotype and phenotype terms, use Punnett Squares to predict, and also doing that pure breeding. So I'll give you guys some coffee beans. Great work today. I'll pull it up the little sheet of the genotype, and I'll close up my Sharpies. But that's up for today. Make sure your names are on those side panels. That's why I'm giving you the Sharpies.